So, welcome everybody to this this presidential lecture, which is the first one this year, and we are very, very happy to be able to present the Professor Bob Holt, or Robert Holt, and from the University of Florida. I think he's an excellent researcher with a kind of living up to what we want to do with these type of presidential lectures, to really, get. And I'm happy that so many are here today also, because it's very important. I think it fits so well into OIST culture that we can be inspired by research that goes beyond and maybe not exactly fits what you are working on, but really but you are prepared in your mind to really think in different directions and above and beyond and in exploratory ways. And exploratory is just what we will listen to here today, and I think it's really interesting to have this possibility. So Bob will share with you his research journey in a way, and then see how he's thinking about the importance of really the experiments in ecology, and also to see this. I would say combine system thinking with the design thinking perspective, and explore that, and also into experiments, not only to see it in observation and theory. I hope I got that right. But we will all see, and we will all be inspired here in a little while. I just wanted to thank you for coming to these presidential lectures, and I hope that you will also think about really interesting questions to ask Bob after his talk. And so with that, I want to give the word to somebody that will present you, Bob, a little better than I just did. And Ulf, here you go.
Thank you very much, Karin, for this lovely introduction and for hosting this presentation. It is such a pleasure to welcome Robert Dan Holt, or affectionately known as Bob to his friends and colleagues here at OIST. Bob is really one of the luminaries of 20th and 21st century ecological theory in North America and in the world. And we feel greatly honored that you came to the small island of Okinawa to share with us your insights and expertise. Bob is renowned for his significant contributions to theoretical and conceptual ecology, particularly in integrating ecological and evolutionary perspectives. If you're not from biology, you would maybe be surprised to hear that ecologists and evolutionists have often worked quite in separation, and in recent years this divide has been mended. The long-awaited marriage of ecology and evolution is really happening, also thanks to genomics, but also thanks to important conceptual and theoretical contributions such as those from Bob. So Bob's contributions have been in the area of population and community ecology, trying to understand patterns of biodiversity and coexistence. This all started already in the 70s with a seminal paper in 77 that often gets highlighted and cited on describing predation and apparent competition as a factor for community organization and determining the structure of ecological communities. Bob has also worked on spatial heterogeneity, so the patterns of biodiversity we see in the real world, and that again involves questions of species persistence, coexistence, and the questions how the traits of organisms that determine their coexistence, their life history traits, are evolving. On a personal note, I have met Bob for the first time around 20 years ago in a workshop I organized to assemble a book on evolutionary conservation biology. And that was meant, again, to bring together ecological and evolutionary thinking with a focus on the application to conservation biology. How does the evolution of organism impact conservation questions? And we invited Bob for a chapter that introduced his very important concept of evolutionary rescue. Evolutionary rescue is a process through which a threatened population that would go extinct in the absence of evolution becomes viable again by sufficiently rapid evolution. And this is an example of what nowadays goes under the name of eco-evolutionary dynamics, in which the timescale of evolution is not fundamentally separated from the timescale of ecology. Many people in the public, in schools, some journalisms I talk with, have this preconception that evolution is very slow, geologically slow. And they think of the dinosaurs, they think of the emergence of Homo sapiens when it comes to evolution. And they have completely not caught up with decades of research on what is known as rapid contemporary evolution. And nowadays, in the times of COVID-19 pandemic, all of us have become familiar with rapid contemporary evolution, with the right selection pressures often created by humans to be strong, and with relatively short generation times, evolution can occur on the timescale of years and decades. And therefore, integrating evolutionary thinking with ecological thinking is not only important conceptually, but also very practically. Throughout his career, Bob has explored a wide array of topics. I already mentioned a few. Let me go over that list again. Competition, predation, disease ecology, habitat fragmentation, spatial ecology, conservation biology, and all aspects of evolutionary biology. His ability to blend theoretical analysis with empirical data and testing has shed light on key concepts in evolutionary ecology. In recognition of his outstanding contributions, Bob has received numerous honors, such as the Sewell Wright Award from the American Society of Naturalists in 2011, the Eminent Ecologist Award from the Ecological Society of America in 2019, and the election to the National Academy of Sciences just a few years ago in 2022. Currently, Bob holds the position of Eminent Scholar and Arthur R. Marshall Jr. Chair in Ecology at the University of Florida in the US, where he continues to advance the field of ecology through his research and mentorship. And with that, welcome again to Bob.
Thank you. Yeah. It's good. Yes, well, it's a great pleasure to be here. It's an honor to be at OIST. It's a beautiful campus. I'd not been here before. I've been to Okinawa. I'm a keen bird watcher, and I joined some ornithologist friends in another part of the island to see some of the endemic birds about 15 years ago. And so I'm glad I did that, because I'm not sure I'd want to go up there in this rainy weather. But if somebody could dim the lights. Oh, thank you very much. Yes, so when Professor Diekman contacted me with this generous invitation to visit OIST, he asked me to suggest something to talk about. So I gave him like 4 or 5, 6 titles, and he says, "Why don't you do them all? But let's pick one that maybe is of broadest interest." And so what I'm going to be talking to you about is actually not theoretical ecology, but empirical ecology, this experimental. A big experiment that I set up as an assistant professor at the University of Kansas that's now 40 years old. It's a big tract of land where we allow different pieces to go into succession, some near a forest, some far from a forest, some big, some small. And I'll tell you about the rationale of that, some of the interesting findings, and why you need long-term studies to really discern lots of important things in ecological systems. But as Professor Diekman said to you, I'm a generalist. I work on many topics, and they fall into 4 areas: predator-prey interaction, indirect interactions, spatial ecology, infectious disease ecology, and the interface of ecology and evolution. And today I'm talking about spatial ecology, habitat fragmentation. But while I'm here, I'm gonna give a talk on each of these other themes, and I hope that some of you. I was just joking that probably the audience is like this, it'll shrink like this every time after you hear me, but hopefully not. And I'm not going to be doing this today. I mean, I'm a theoretical ecologist, and even theoretical ecologists don't like to hear talks that are full of equations, basically, okay? Unless they're masochists, okay? Instead, I'm going to talk about organisms. These are all organisms that were important in this field site that I'll be describing for you. It's just more fun. So this is an experiment that we started in 1983 and is still running at the field station of the University of Kansas, and here's a false infrared false color image from the air of it. And what had been an agricultural field let go partially into succession. We mowed in between the patches. I'm going to give you a little personal history that'll be intertwined with intellectual history of what motivated this experiment. And then we'll walk through the design of the experiment and some of the interesting patterns that we've gotten in the past up to the present. Why should we even care about history? William Faulkner, in his great novel, Requiem for None, said, "The past is not dead, it's not even past." The concepts that guide our thinking come often from deep history in our disciplines and even outside our disciplines, and being aware of that, I think, is important. So I'm giving you a little personal history to start with that'll be tied in with the history. So my background is I was a physics major at Princeton, but I was interested in biology. I thought I was going to be a biophysicist at first, but I was a very keen naturalist. I loved being outdoors, and I did field work in the summers in Mexico, in the Caribbean, in New Jersey, and I took biology courses for fun, so by the time I was a senior, I could go to graduate school in biology rather than in physics. Robert MacArthur, who was a very famous ecologist, for those of you who are not in ecology, was my undergraduate advisor before he died. I went to Harvard, and then I did theory, and then I did some field work as well. I moved to Kansas in 1979. They hired me as their theoretical ecologist, and I was the only one for at least 500 miles in any direction. And the problem is, you would never have graduate students because we didn't have a critical mass of people to attract them, competing with Princeton and Stanford and Cambridge and so forth. And so I said, I've got to do something that's empirical, not just theory. I'll just be by myself in my office writing papers. So I was interested in the tropics. So I started out by doing field work in Cozumel and other islands on Anolis, continuing what I did a little bit in grad school. And I was a keen bird watcher, and I had some ideas about mixed species foraging flocks of birds. And these were really good ideas, but in the course of doing them, I realized to do them right, I would have to live in the tropics like half of each year for many years. And by then I was already married, and my wife had no interest in being in, Amazonian Peru with me 6 months in a year. And also getting the data was painfully slow. And there were people who were better at it than me. Okay? So I needed, by the early 80s, I've been hired in 79, I knew I needed to find something that was local, not in the tropics, i.e. somewhere near Kansas, and that would also work, okay? It's very easy to come up with great ideas that when you actually get out in the field are really difficult to do and require an entire lifetime to do right. And I didn't have an entire lifetime. So the group of ecologists who were working on the field station got funding from the university to set up some kind of experiment out there, because that's what you can do if you have land that's owned by a university. You can set up experiments that maintain integrity over long periods of time, so you don't have to think over just 2 or 3 years, but decades. And so what is a classic ecological idea that requires a long time scale to get a handle on? It's what's called succession, which is after disturbance, say the abandonment of a field, a glacier that retreats, you see a system that starts from some beginning state and goes through a sequential series of replacements of species, ecosystems, habitats, and so forth, which is sort of how ecology started with Clements and Cowles and Gleason and people like that in the 1890s, early 1900s, and it still goes on today. And then we were going to set up an experiment that had to do with how does the time that you initiate the process, start the disturbance, influence the successional dynamic? Perfectly reasonable empirical question. We got the money, and I was the only assistant professor, the only young person in the department entirely made up of tenure faculty, and this was very risky. I was thinking about it, I said, there's a problem with this. One is try to set up we've got one field, and you're going to be setting it up so part of it goes out in the winter, part of it in the summer, but the one that goes out in the winter, it'll start seeding the one in the summer, so there's a confounding in the spatial design at the get-go. And there were no clear a priori hypotheses about what might happen. You thought something would happen, but saying why. So there were two problems. So I persuaded my colleagues to go in a totally different direction, which I think was a little bit risky, which was the interplay of habitat fragmentation and secondary succession. So if you think about habitat destruction and fragmentation, this is one of the major drivers of the extinction crisis. You know, the Midwest, where Kansas is, the native prairies are just little tiny pieces of their former vast selves. And, you know, that's true for most habitats around the world, forests and so forth. And there's a huge literature on habitat fragmentation, many observational studies, lots of models. There's only a handful of experiments. A postdoc and I, Diane Dubinsky, who's at Montana, over 20 years had a review in conservation biology, and we could find only 20 such experimental studies where a landscape was manipulated to study habitat fragmentation worldwide, and there aren't any more out there today. And the reason is it's hard to do landscape experiments. It takes a lot of land, a lot of people, a lot of money, so forth. So what's a habitat fragmentation? It's a deliberately created spatial pattern of patches in a landscape. It's as simple as that. And at the time when I was having these ideas, there was exactly one such experiment in the world created by a great conservation biologist who sadly died about 2 years ago, Tom Lovejoy. And he had told me about it, and I think the very first paper had just appeared, and it's near Brazil. And Tom was concerned with destruction of tropical forests. And Brazil had just passed a law that ranchers in the Amazon had to leave behind a fraction of their land in forest. And he persuaded these ranchers near Manaus, Brazil, to leave patches behind in an experimental design where you have continuous forests, but you have big fragments and little fragments, and you look at, say, these, you know, understory ant birds and see what the impact is over time. So here's just another aerial shot of one of his study areas. And that started in 1979. So what was this conceptual framework? So I'm going to walk through kind of the ideas here that kind of motivated my experiment. It was what's called island biogeography, which is, if you can think of habitat patches, it'd be like islands in a sea of a radically different habitat, just like Okinawa is an island in an actual sea. And island biogeography focuses on spatial attributes of the island. How big is it? How far is it? You know, how far is Okinawa from Asia, for instance, from the mainland of Asia? Because that's where colonists are likely to come from. And how does that influence dynamics? And MacArthur, who had been my undergraduate advisor, remember, and E.O. Wilson published a very famous book, 1967, Theory of Island Biogeography. They worked this out. And so I'd been influenced by these people. Wilson was one of my professors at Harvard when I was a grad student. So what they were concerned with, like in the Florida Keys, you got some big islands that are near the mainland, some big islands that are far from the mainland, and little islands that can be near and far. And that you might be interested in, how does species richness vary with the size of the island you're looking on? So these are birds on an archipelago in the Baltic. These are ants in Melanesia that Wilson did, the sort of things that Evan Economou was interested in, right? And MacArthur Wilson came up with this very famous conceptual model, which you can mathematize in various ways, which looks at how does a community rise? All local communities are dynamic, and over some long time scale, every species you see in a local community came in from someplace else, and there were species there in the past that have gone extinct. And so that dynamic process of colonization extinction has to be explaining in some way the diversity of species you find. So MacArthur and Wilson said, you've got some source area, say Okinawa has been wiped out by a tsunami and everything's starting to come in from Asia. And finally, everything that can possibly come in comes in. And so colonization has got to go down. But the more species you got, the greater the extinction rate, which would be, and they start interacting with the other and so forth. And then you reach an equilibrium, and the equilibrium maybe the extinction rate's higher on small islands than large islands. Voila, you get the so-called species-area relationship emerging from the dynamics of colonization and extinction. So the real fundamental insight of MacArthur and Wilson wasn't notions about equilibrium or whatever. It was that you have to view communities dynamically in terms of colonization and extinction. How does this relate to habitat fragmentation? Well, this is how they ended their monograph, where this is very famous work by Curtis, a plant ecologist in Wisconsin. This is a tract of land that in the mid-1800s is all forest, and as agriculture came in, there's less and less of it there, and what's left are little bitty bits, okay? And what do you expect to happen? Here you have, if you went out in that continuous forest ahead of time, you could take samples of different sizes. You can hardly see them in this figure here. And you can say, how many species do I have as a function of my sample area? And obviously, the bigger your sample, the more species you're likely to catch. But then you fragment it, you destroy a lot of habitat. What you're going to see is you're going to start seeing extinctions and reductions in colonization. And what was expected was that extinctions would be occurring, but occurring faster on small islands because population sizes are low. So you get a steeper area relationship emerging over time in the fragmented landscape, okay? Now that was one massive paradigm in ecology having to do with community assembly via colonization and extinction. And it occurred to me that there was another classic paradigm in community ecology that also involved colonization and extinction, which was this issue of succession. A farmer feels abandoned. You get short-lived herbs and grasses and come in for a while. They get replaced by, so you get these waxing and waning of species numbers, long-lived herbs, finally woody plants in the eastern U.S. show up, and eventually it'll be a forest if you leave it alone, right? And so you get this successional dynamic that involves what? Recurrent colonizations and extinctions, whatever's there. Well, one of my professors at Princeton was Henry Horn, who developed a lot of Markov models of forest succession. So I had succession kind of in the back of my head too. I said, well, why don't we put these two things together? Kind of the ideas from island biogeography and the ideas about succession. Because nobody had asked, and then succession occurs also in entire food webs, not just plants. So this is a classic idea about succession. You get some species that's very good at colonizing, but maybe is short, and it has little bitty seeds, and some more sluggish colonizer shows up and replaces it because it's a superior competitor, but it's a slower colonizer, so you get a successional sequence. The same thing can occur because of food web interactions. This goes back to the paper that Ulf was talking about earlier, where a species shows up, but then a herbivore shows up, and then alternative prey shows up slowly and sustains that herbivore as that first species goes out. So there were this is in the early 80s, two questions that were largely unanswered, not even asked, about succession. Are there area and distance effects in succession, just like there are in island biogeography? And then is trophic dynamics driving it? Now, this is something I'm very interested in, but I'm not going to say very much about it because it turned out our data had to do mainly with plants and a few consumer groups, not entire food webs and so forth. So the conceptual model for this experiment was that you're going to change something like patch area or distance, and that could actually affect lots of things. It could affect colonization and extinction dynamics of the plants. It could affect ecosystem processes. Maybe small patches are drier, more exposed to the wind or whatever, and it could affect how many herbivores are present, for instance, to drive successional dynamics. And so one of the fascinating things and complications that's challenging in these landscape experiments is that when you create a landscape, you're changing lots of things simultaneously because of your enforced spatial design. So there could be lots of causal processes at play underlying any interesting pattern you see. So you can see lots of patterns, but actually nailing that exactly why it's happening is harder. And I've done a lot of theory that's related to some of these ideas that I'm not going to be talking about. And now if we're talking about succession, we could be talking about two different scenarios. So what I was thinking about was like island biogeography, where you have patches that are isolated in middle of a farmer's field, and you mow in between them, and so you're looking at succession on these little patches. Another option would be you're looking at a giant tract of forest, and you clear-cut patches of forest, and you got big patches that you clear-cut and little patches, and succession might take place very differently there because on those patches, clear-cuts in the forest, little patches are very near. There's no distance between any place in the patch and the source area for seeds, but in a huge clear cut, most of it may be far away. Whereas I expected for various reasons that in isolated patches, the rate of succession ought to be faster as a function of patch area. And also, like I said, there's this issue of tying food web ecology in here. I think I'll just sort of skip through this because that's not what our data tended to be about, okay? But very interesting. So back to Kansas. What motivated the experimental design? So here we have a schematic of the field, and to the west and the south is a large tract of forest with kind of fields mainly to the north and east. And we put patches of several sizes, and we're separating them all by intense mowing, okay? And then we're going to let the little patches go through succession and the big patches. And the big patches are 50 by 100 meters, so that's half a hectare. And these are 4 by 8. And then we've got different distances from the forest, and we try to have replication and interspersion of treatments, okay? And then I'll explain why this sort of an unusual design. We've got these little patches laid out in these clusters. And one reason is we wanted to make sure that we had the same amount of sampling in small patches as large patches, which is what this design does. These little dots here, they're sort of the sampling stations for plants, for insects, for mammals, and so forth. Here's a fairly recent Google Earth image, for instance, of the site. And the idea here is you have here 50 by 100s, and you've laid out your plant quadrats here. And things can move around easily inside that. And then we're going to have a cluster of small patches that collectively span the same amount of the Earth's surface. That means whatever heterogeneity happens to be present in the soil or in the initial seed bank or whatever is going to be the same on average between large patches and these clusters of small patches, okay? But these are not connected to each other, right? So things can't move easily between them. And I did some theory, and I'll go through this very quickly, saying that internal connectivity inside the big patches could matter a lot. Because you look at a little patch, it could only be colonized from outside, but a sample area inside the big patch could be colonized from outside, but also from the surrounding area, and being coupled to the surrounding area could also affect local extinction rates and the like. And so I did some theory suggesting that was the case. So I'll go through this quickly. So this is what our study area looked like in 1984 when we first set it out, okay, from the air. And I think it's actually a really pretty picture. I ought to put this up on my,
It looks like a painting.
It looks like a painting, yeah. In fact, I've been joking. I like modern art. I like a lot of art. And I don't know if you know the Dutch painter Mondrian, who was famous for these squares and lines. This is Earth Mondrian, right? Now, that was 1984. Today, now it's 2025. That experiment is still out there. It is one of the longest running experiments in ecology. So the Manaus study, so we had a paper in Science Advances a few years ago that brought together the long-running projects. The Manaus project is still out there. It was the earliest, 1979. Ours started up in 83, 84. And then there are others in Australia and various other places in the U.S. and in Borneo going since. But there aren't very many of them still. Now, I left Kansas in 2001 to take on this endowed chair at the University of Florida, and we had a hiatus in data collection. So I'll be telling you some about what happened those first 20 years, and then we've had a spurt of new data that's come in when we've got some grant funding. But we got a lot of information over time. The plant community over that period, we had over 300 species, 80,000 records in these permanent quadrats. When trees started colonizing, we started tagging them. We tagged 8,000 trees out there and many, many repeat measurements of individuals. We had insect ecologists did some work, and then we had very detailed work on small mammals with lots and lots of capture. So it's a very large data set to allow you to see patterns. This is what it looked like early on. This is what's called an old field. It's just you got what are called asters and goldenrod, sort of classic herbaceous plants and grasses that dominate the field. We have these permanent quadrats. Here Henry Fitch, herpetologist, postdoc of mine, George Robinson. Here's a small mammal ecologist, Mike Gaines. Here's a snake trap that was put out by Henry. Snakes like to worm themselves up under these pieces of metal. And 8 years later, we got a cover in Science. And I think we got the cover in Science because of a sort of a paradox. We had lots of interesting patterns at the level of what's happening to individual species, and I'll show you some of those in a second. But there was no overall effect on succession. Remember, that was my original prediction that there would be, okay? I thought things would happen really fast. Here's some of the patterns, okay? So here are the small mammals. So here's a deer mouse, 20 grams, cotton rat, 160 grams. And these are the different patch sizes. Here's the big patches, the medium patches, the little patches. Cotton rats were almost only on the big patches. Deer mice are actually quite common on the small patches. Now, maybe that involves competition or something else going on, but there clearly was a sorting out in the mammal community by patch size. Here's snakes from Henry Snake's traps. He went out and just said, how many times do you find a snake under one of those pieces of metal? This is the number of snakes per trap on a big patch versus small trap. Okay, there was a patch size signal in the snakes. Well, these snakes all eat small rodents. Maybe that's another reason why this rodent is. So this is where, we can come with lots of hypotheses for why this pattern occurs. Actually, to nail them down would be challenging. There was also, these are plants. You go back to the same meter square quadrat 1 year, and the next year say, is plant species A still present? Did it persist? Well, it turns out that that's true. Colonial perennial plants persisted better on big patches than on small patches. Annuals, there was no effect. Okay, so there were these signals at the population level, but not at the whole system level. I think that's why we got into science, actually. Okay, plus we had that pretty picture for the cover. And then if you look at species richness, remember, had to do with island-wide geography. Yes, big patches had more plant species than small patches, but if you look at the number per unit meter, number in your quadrats, this line here is an actual regression line. That is the flattest regression line you'll ever see in ecology. There's no signal of being in a big patch or a small patch at a very local level, okay? We're going to see that this changes as time goes on. And as time goes on, what's happening? That initial feel, that first 8 years, what had not occurred to me was that outside colonization would be so slow. What we were seeing was a reshuffling of species that were all there in the first year in the seed bank. And some species came up and they gradually grew and dominated other species competitively and the like. But as time went on, we started having colonization from the woodland into our patches. This is the late 1990s. You can see these little trees that have started showing up out there. And here's a field crew, and this is roughly dogwood. I'll be showing you in this. Brian Foster is one of my great colleagues, plant ecologist. And so succession really did start taking place. So early on, it was annual species, perennial forbs, perennial grasses. But woody plants started coming in after the first 10 years and just grew and grew. And the cover of perennial forbs, herbs, grasses, and the like went down, okay? So let's just see. So here's an example. 1984, this is what it looked like when we started out. Here it is 12 years later. You can see these little woodlands that are starting to develop, all right? And here we are. We built a tower so students could study foxes and other mammals out there. But I took a shot looking towards the woodland from that tower. And even to my naked eye, these small patches are near the forest, but there are fewer trees on them than these big patches that are further away. And by golly, you go out and you measure it, and what we did with the trees was we looked in 4 meter by 4 meter quadrats and counted just the number of trees greater than 2 meters in height, okay? And what you see here is by 1990, remember we started in 83, 84, 10 years later, trees started coming in, and over time they just grew, okay? So this is adding up over all species, and as they grew, there were more trees, individuals, on big patches than in small patches per unit area. And also there were more trees nearer the forest and further from the forest. So here are some pictures. They're colonizing, there was a lag in their colonization, but there are more of them here in the big patches than the little patches. Why? Okay, well, we get a handle on this a little bit. Is it because a big patch is a better place for a tree to be? Okay, well, we could get at that a little bit. Because we could go out and as trees came in, we could put marks on them and go out over time and measure their survivorship, their growth rate and height, and their fecundity for these rapidly growing early successional trees. And this shows, for that species that I was just showing you, the rough-leaved dogwood, I was showing the data before was everything added up over all species that was greater than 2 meters in height. And there is a patch signal there. This is looking at the smaller size classes, going smaller and smaller here. And notice this huge bulge of young individuals that are showing up in small patches. This is per unit area versus large patches, okay? Now, is this because it's a better place to be a tree? Well, you can go out and do demography and get a handle on that. So you can look at fecundity. These dogwoods produce clusters of fruit. Count up the clusters of fruit. look how you have a covariative density you have to worry about, because you always expect density dependence in plants. And controlling for density, fecundity per tree is higher on small patches than large patches. So it's a better place. Small patches are better than large patches in terms of reproduction. You can do the same for survival, same for growth. Put them all together in what's called a stage-structured transition matrix model. Look at the long-term asymptotic growth that emerges from that model. And controlling for density, it's not the case that small patches are worse places to be. It's better to be in a small patch. Why? If you're a tree, you don't have competitors around you, for starters, okay? You're getting all the light. You're not being shaded out by your competitors. So we can reject the hypothesis that big patches are better places to be a tree than small patches. What does that leave to explain the pattern? Well, what we think is that it's not because of differences in patch quality, it's because of how patches are coupled to the larger landscape. So how do these trees disperse? Many of them, like the dogwood, are bird dispersed. And birds, we did some work on this, seem to orient more towards hanging out in the big patches than small patches, and so that they're more likely to bring in seeds from outside and moving around inside the large patches, okay? So it's kind of an emergent regional spatial effect that explains the patch size signal and successional that we're seeing. There's probably other things going on as well that I won't get into. So that was a little bit about the plants. We're going to come back to plants. Let's talk about the mammals for a while. We had about 8 species of rodents, but these are the ones that were common enough to make something of. Here are some of the mammalogists who work there doing what mammalogists do. So cotton rats, which are the largest ones in our community, every time we caught remember, and these are individuals that you're following around. You're catching them in traps, and every time you found the same animal in 2 traps, you put a line down connecting the traps on a map. And so this is all those movements from the traps of cotton rats, and they're moving inside the big patches a lot. They're moving between the big patches, but they're hardly showing up the small patches at all, okay? And in fact, the small patches seem to be sinks. They were all kind of subadults that weren't reproductive. But over time, it was interesting. I told you early on in that science article, cotton rats were almost entirely in the large patches, but as time went on, they started using the other patches more, associated with the change in succession. So there's a relationship between the spatial distribution of the species, what kind of patch size you find in, and the vegetation stage of that stage. Now this is one of the smaller species, the prairie vole, which was found in all the patch sizes early on, but as time went on, it actually was found most commonly in the small patches. And we concluded, I had a student who did a detailed study, this had to do with behavior. What they did was the patches were now dense enough, they provided good places to hide, good places to build a nest, but they could forage outside in this highly productive mown area, okay? And so they were shuttling between these 2 habitats to sustain a much larger population. And this is their movements, okay? Very, very different from the cotton rats in the same field. Then finally, a woodland species colonized. It was totally absent early on. It shows up. Remember, woodlands are better developed on the big patches than the small patches, okay? So that all makes sense. Then finally, we had a mystery organism show up, the wood rat, which is a huge, huge rodent, which make these giant middens. And they had been totally absent. I mean, we found 2 individuals out of those 38,000 captures in the first 20 years of the study. We went back 2015 and found a bunch of these mounds in this very thick vegetation. So this is a late colonist. And there are reasons that nobody has recognized this. So Bill here is 6 feet tall, so you see these mounds. But they showed up, they exploded, and then they seem to have disappeared about 10 years later. These are my current collaborators out there. So I'm going to show you a few more plant patterns. So remember, we have these early successional species that show up, and I should have said that all these species, we have almost no invasives. They're all natives of one sort or another. So we have all these lovely early short-lived perennials that show up. They're all going to go extinct because of succession towards woody vegetation, all right? These are things you find in open fields and prairies and so forth. So how does extinction take place in this system? Remember, that's part, you've got colonization and extinction driving community assembly. We know that the later successional plants, the woody species, are colonizing faster in larger patches, So they ought to linger longer on the small patches, okay? But the small patches had small population to begin with, and just like island biogeography, you might expect extinction rates to be higher there for that reason. So you have two alternative hypotheses here, okay? And so what happened? Well, first place, what's the data look like? We have all these permanent quadrats out there, right? And we aggregate all the data from the large patches, all the quadrats there, and then the same rough number of quadrats in the small patches. And then we're going to compare big to small patches. And there are two ways to characterize a population in a data set like that. You have what fraction of the quadrats are occupied. That's its distribution in space. That's what's called occupancy. And then you have abundance. How abundant is it within an occupied? And both can change over time. And so we needed to come up with something that used both kinds of information. And what I came up with was kind of a graphical way of illustrating what's happening that shows patterns. You could do some statistics. You almost don't need statistics. So we take our species and we just put it in one data set. And then we rank order from the most abundant quadrat to the least abundant quadrat, include all the zeros. So this is rank abundance. This is all the data, okay? What we call the rank abundant profile. And this is showing that same procedure for all the quadrats as follows. Okay. And you can do some randomization tests and so forth showing that the minor differences there are not likely to be significant. So what happens over time? This shows that same pattern. It's an aster, a lovely perennial. 1985, large versus small. I just show you those are not the same. As time goes on, it starts going extinct. It's declining in both its maximal abundance and in the fraction of the field it occupies. And in doing so, notice that it's going extinct faster on the small patches than it is on the large, even though it's going extinct overall, okay? Pretty intriguing pattern. So this shows it in a map. Each bubble here is a measure of local abundance. This is the abundance across the field in 1985, and by 10 years later, it's clearly disappearing from the field, but where is it hanging out? It's staying in the big patches rather than small patches. And so you get this effect that even though succession is taking place more on the big patches, this species that's being driven extinct is still persisting better there. But other species show the opposite pattern where they actually persist. Common ragweed, which is a notorious pest for people with allergies in the U.S., actually persists better in small patches. And it turns out that if you look at the patterns of extinction, you can have some species like the aster decrease in occupancy while it was decreasing in abundance. Others stay widespread. They just decrease in abundance everywhere. Some species actually get more widespread even though they decrease in abundance and vice versa. You get all these different spatial patterns occurring, and figuring out why is challenging. And the only thing we could find in the life histories of the species was that the species that persisted in large patches had clonal reproduction. They had vegetative reproduction, which allows them to do this sort of short distance colonization via sending out runners and so forth. Whereas the species persisting better in small patches were all annuals, entirely dependent upon seeds for recruitment. Maybe there's more disturbance in the small patches, for instance, and they can deal with that better. Maybe there's relaxed competition for clonal species. We don't really know. But you have all these species-specific stories out there on how extinction is taking place, even though the general message is that you have a whole suite of early successional species that are going extinct. How extinction occurs differ species by species. So this fragmentation overlays other kinds of spatial heterogeneity. This is a farmer's field. There was terracing. There was probably little differences in how much nitrogen was spread around everywhere. How do we understand the overlay of the fragmentation effects with other kinds of causes of what's called beta diversity? That's turnover in species, composition in space from one site to the next site. And what we expected to happen was when species first show up, they're showing up for reasons that are only loosely related to the local properties of the site. And they ought to be like they show up at the west end of the field or they show up on the north end of the field. And it'll take a while for environmental signals to show up, sorting out by how much silt is in the soil, for instance, and so forth. But the environmental signal we expected to be stronger through time. This is a paper we had in Journal of Ecology a few years back. When we look at all the quadrants and large patches, do kind of an analysis of variance, the fraction of the variance in composition among patches that's explained by pure environmental variables grows, and the spatial signal gets weaker. But if you look at the small patches, the spatial signal, if anything, gets stronger over time. Like there may be a priority effect. Something shows up and then just dominates a little patch, for instance. Okay? Succession precedes apace. So here's what the site looks like in 2009. Okay, 2015 from space, basically. We got little woodlands out there. These are what the fragments look like from the ground. They're little forests. What happens to the plant community? Well, one of our former grad students is now a professor at Bard College in New York, and she got a very nice NSF grant to go out and resample all the sites, which he did over several years until COVID hit. So this is looking at what's happening in a square meter quadrat over time, okay? And we separate patches that are large and far from the pot. So they're patches that are large, they're patches that are small, and so forth, okay? Patches that are, the red ones are large patches, the blue ones are small patches, the open ones are near the forest, the closed ones are far from the way. So as time went on, I left Kansas around here to go to Florida. Species richness is increasing, and by the time I left, there was beginning to be a very subtle signal that there were more species at that very local meter square scale in big patches near the forest than, say, small patches further away. But it's nothing that you go sort of ho-hum. It's a very kind of minor effect. Well, Kathy went back and resampled it, and if you went to a quadrat that's in a small patch far from the forest, there's about 8 species on average. There's twice as many on the big patches near the forest. So the spatial signal on species richness is now huge compared to what it was initially. And we're in the process of analyzing all this data because there's a lot of other interesting data as well. So the edges of the large patches are sort of in between the interior of the large patches and the small ones, for instance. Well, this is actually a really miserable place to work. If you look at the studies of deciduous forest and succession, they tend to be bimodal in the time periods they work. People work on old fields. We have all these pretty herbaceous plants and so forth, or they work in mature forests. But there's this zone in between where woody plants have shown up, but they're all close together, and it's very thick. And there's a very scant literature on it. And that's because it's a miserable place to be. And in the eastern U.S., what you find is what's called poison ivy, which is a plant that is toxic to touch. You find brambles, you find lots of ticks. And so this is Kathy in a hazmat suit in a Kansas July out there to get this data. This is why there's very little data like this in the literature, because it's the kind of habitat that's miserable to live in. But nonetheless, we've got some really pretty interesting patterns. So the pattern that I just showed you matches what I expected from kind of island-based geographic theory, that finally species richness is greater at a local scale on big patches than small. But just because that pattern is consistent with that process doesn't mean that's what's necessarily going on. And I'm not going to tell you what the current hypothesis that Kathy, colleagues, and I are exploring, but there may be more subtle things going on involving how patch size influences local species coexistence mechanisms by modulating things like temporal variability and so forth, okay? It might not just be the sort of island-wide geographic effects of colonization and extinction. So conclusions from this study, it took a decade. I mean, I got a paper in Science with null results. That takes some effort, okay? That's because I think, as I said, it was contrasting the null result at the macro scale to the really interesting patterns at levels of individual species. But finally, the prediction I'd made that patch size and distance would have an effect on succession showed up, but it took a decade for it to show up, okay, because colonization was just sluggish from the outside. And it's still ongoing. There's species of trees that are found in the woodland nearby, oaks and hickories, that are just now starting to show up in our site. And so there's an ongoing temporal process that's still unfolding. for the patterns and species heuristics to show up at the very fine spatial scales of meter square quadrats, it took another decade for that to show up, okay? Then the effects of fragmentation on particular species, like the rodents I was talking about, vary greatly over time, reflecting a kind of complex interplay of spatial structure of the environment and the kind of vegetation that's emerged. And we think that the things like trophic interactions, predator-prey interactions, competitive interactions, and the like are also at play here. And so there's just a lot of interesting species-specific stories out there where we now have lots of patterns, but actually coming up with nailing down the processes is sort of the next generation of work. And my general conclusion is that many ecological processes that underlie the patterns you see out in the real world involve very long transient behaviors. And so they're going to be missed in short-term or snapshot studies. And that's one thing that institutions can do, is help set up, like Canvas did in its field station, sites that have long-term institutional integrity where people can go out over multiple generations and gather data from the same site, because they will start seeing things that you will always miss in short-term snapshot studies. And there's no way, I had hundreds of collaborators, lots of students suffered out there, I think. Here's some of them. Does anybody know what they're doing? Tick search, exactly. And we had funding sources, and and that's where I wanted to stop, okay? So I want to leave a little time for questions. So thank you very much.